I think a lot of us have the intuition that simply scaling up the current approaches will not take us all the way to where we might want to be. I shy away from the term AGI. And so the hypothesis in that paper is that the reason we don't have a longer genome is that it's actually a feature, not a bug. So my answer is that Lamarckian evolution is a much better algorithm, and that's how things should work.
Hi everyone, welcome back to the Embodied AI podcast. You just heard the voice of Tony Zador. Tony has a lab at Cold Spring Harbour in New York. In his lab, he studies auditory decision-making in rodents, does some work on molecular connectomics, and uses insights from neuroscience to inspire AI. At the start of the episode, we have a little detour on language and the Costa Rican singing mouse, but then we turn to the Embodied Turing Test, an idea that was discussed in a recent paper by him and 27 other pretty influential people in AI. In the paper they discuss Moravec's paradox, which is the idea that what we humans find hard is actually quite easy for AI, and the things we find easy, like sensory motor capabilities, are very difficult for AI. In the paper they also discuss the possibility of one or multiple platforms where non-human animal-like virtual agents could be trained to pass the Embodied Turing Test. We also discuss Tony's work on the genomic bottleneck and how it might help in building AI that is better at generalization and transfer learning. We also discuss his work on molecular connectomics. At the end, there's also a sociological question about how different people think about evolution and the idea of quote-unquote human uniqueness, and as well as some career questions. As usual, timestamps for the questions and all the papers we discuss are in the description. Enjoy. Hi Tony, it's great to have you here. So let's start with a question about your background. The last two episodes were on language, and let's stick with that for a second. So you did a bachelor in linguistics, and do you ever connect what you learned back then to your current work on auditory decision-making in rodents or the NeuroAI work?
Yeah, it's funny that I have a degree in linguistics because I haven't really done much with it. When I started my undergrad training, I actually started off in physics because I enjoyed math and physics and liked solving math problems. I liked rigorous solutions. But at some point relatively early on, I realized that much as I loved solving for how fast the ball rolled down the inclined plane, I really couldn't care less about the answer. You know, I sort of had been reading ahead in what the big questions in physics were. And they were questions that ultimately that other people are working on, but didn't really move me passionately. You know, what was happening on length scales, you know, either way smaller or way bigger than anything that I interact with on my daily life. And I guess also what was driving me was the realization that our perception of reality is as much due to our perception as the reality. And that got me interested in thinking about thought and how thought emerges from matter. The thing is that this was in the early 80s. And although there were neuroscientists around, I didn't know about them. I kind of, through introspection, concluded that the key to thought, the key to consciousness, which were the things that I found really interesting, were in language. And I no longer believe that necessarily. In fact, I believe just the opposite. But at the time, that was my intuition. And so who studies language? Well, linguists study language. And so that's how I sort of moved from physics to linguistics. The kind of linguistics that I was doing was very formal, rigorous, sort of generative linguistics, which had some of the flavor of physics. So my undergraduate thesis was on generative phonology, and it was about coming up with a set of rules to describe a particular linguistic phenomenon, the phenomenon of vowel epemphasis in Hungarian. So that's how I got into linguistics. And then by the end of it, I realized actually what drove me away, it was actually the formality of linguistics that I was studying, the absolute lack of connection to the mechanism whereby we produce sentences or sort of instantiate these sets of rules. And that was how I realized I didn't want to go on in linguistics. I still hadn't discovered that there was this field called neuroscience. So I sort of wandered around for a while. So that interest in language and linguistics has always been in the back of my mind. And really only in the last couple of years, it probably had a little to do with my choice at some point to go work on the auditory cortex rather than the visual cortex, which is where a lot of the work in animal neuroscience is. But obviously, since I was working in animals, it didn't seem very plausible that we would be studying language in animals. And really only in the last couple of years with the rise of large language models and my interest in them, has any of my linguistic training seemed at all even slightly relevant.
Yeah. I think it's really interesting though, because clearly we wouldn't like attribute language to animals because we normally relate it to like symbolic things. I think I only like found out maybe a year or so ago that mice did these like high frequency vocalizations we can't hear. And I guess cats can hear them. When I thought, I was like, auditory decision making language, or is there like a link to that? Because I guess the task you're looking at wouldn't be very language based, but I always thought that it'd be interesting if you were sort of long term trajectory thinking, maybe there's something interesting. Yeah.
Yeah, so it's funny. There are certainly quite a few animals that vocalize. In fact, one of my newest colleagues at Cold Spring Harbour, Arka Banerjee, studies a particular species of mouse, the Costa Rican singing mouse, that is so named because it actually has stereotyped vocalizations in the human auditory range. It's actually, I don't want to say unique, but it's very rare among mammals, especially non-primates, that it produces these very elaborate stereotyped vocalizations, sort of almost like songbirds. And they use them in some way to communicate. But what interested me about language was not just acoustic vocalizations, but as you say, symbolic communication. And I'm fairly confident that no animal, and certainly not the Costa Rican singing mouse, generate, sophisticated vocalizations as part of symbolic communication. So basically, you're stuck to studying humans if that's what you're interested in.
All right. I'll get back to language actually in a second. So let's talk about the Embodied Turing Test, which I think just sounds really cool. I'm maybe a bit biased considering the podcast name, but so there was a recent preprint called Towards the Next Generation of AI, Catalyzing the NeuroAI Revolution. And besides it starting a really big Twitter discussion, which was interesting, there were a lot of like 28 people were involved as authors and a lot of big names in AI. And I was just wondering, you as a lead author, what was the process of planning and writing up a paper like this?
So if you Google herding cats, that gives you an inkling as to what the process was of trying to get 28 authors who sort of agreed on the basics, but not necessarily on the details. The particular list of authors came out of a meeting that I organized back in 2020. In fact, it was going to be in March 2020, but if you think back to that time, you can imagine why it wasn't held in March 2020 at Cold Spring Harbour, which was called NICES. And it was really an attempt to bring together people interested in neuroscience and AI, and specifically people interested in applying insights from neuroscience to AI. A large fraction of them shared this vision that although what's going on right now in AI is mind-bogglingly impressive and the rate of progress is like nothing I've ever seen, and I think unparalleled perhaps in the history of science and engineering. At the same time, I think a lot of us have the intuition that simply scaling up the current approaches will not take us all the way to where we might want to be. I shy away from the term AGI, artificial general intelligence, because I don't think intelligence is general. But I think many of us want is AHI, artificial human intelligence. And it's not clear that the current approaches, impressive though they are, will get us there. So what's missing? What I think many of us think is missing is really captured in Moravec's paradox. So Moravec I've never met him, although I'm a fan of his work. It's a computer scientist. He wrote a number of books, but come to be known in part for the paradox that's named after him, that he really articulated nicely in a passage that I maybe will take the liberty of reading. He wrote, "Encoded in the large, highly evolved sensory and motor portions of the human brain is 1 billion years of experience about the nature of the world and how to survive in it. The deliberate process we call reasoning is, I believe, the thinnest veneer of human thought, effective only because it is supported by this much older and more powerful, though usually unconscious, sensory motor knowledge. We're all prodigious Olympians in perceptual and motor areas, so good we make the difficult look easy. Abstract thought, though, is a new trick, perhaps less than 100,000 years old. We have not yet mastered it. It is not all that intrinsically difficult. It just seems so when we do it. So what he's saying there is something that many people have also recognized, which is that what is often easy for computers is hard for humans and vice versa. So obviously, computers beat humans in chess in, I think it was 97, when there was the famous match between Big Blue and Kasparov. But if you ask them to play chess, they'll struggle. They'll be unable to pick up the chess piece. So the stuff we take for granted, they can't do. And more recently, AI has really begun to make tremendous progress, not only on the things that are difficult, that we think is the pinnacle of human achievement, but even the things that are uniquely or especially human, like language. And I think that's been the breakthrough recently with large language models. But they haven't, as Moravec put it, mastered sensory motor knowledge, which turns out to be incredibly different and goes beyond just robotics. It's knowing everything about the world that is not purely accessible in rule-based, in fact, linguistic perception. And I guess the idea is that there's probably what you might call the dark matter of knowledge, the stuff that is just not available, even if you suck in and digest all the words and sentences that have ever been posted on the internet. So that was really the process. There was this agreement by, all the people who signed on to this document. That's really the challenge going forward. And also, I think more generally that neuroscience has a lot to offer AI, and that the history of AI is really the history of taking ideas and insights from neuroscience and then shaping them with, sophisticated engineering, turning them into something useful.
Yeah, I suppose if you ask Jan LeCun, the dark matter of intelligence would be self-supervised learning. But so let's go back to the large language models you mentioned, right? And papers about the embodied Turing test. And as you said, language is sort of traditionally thought as the pinnacle of human intelligence. And I guess that's why Turing, when he came up with the Turing test, he did it sort of through the language medium. If you look at current transformers, they seem really impressive. How could they still, in theory, maybe fail the traditional Turing test in terms of convincing us that they are human?
Well, so there are two questions, right? First, how would the large language models fail the traditional Turing test? And how would any system fail the embodied Turing test? So I'm an outsider. I don't work at DeepMind or Microsoft. And so I don't get access to the current generation of large language models. I get to play with GPT-3. GPT-3 is incredibly impressive, but there's little doubt that it's not quite passing even the standard Turing test. Although I think perhaps for somebody who hasn't really played around with it much, it might do a pretty good job. It's sort of a weak test because, even when I was a kid, there were these very simple systems like ELIZA. I don't know if those are still ever trotted out, but, the psychotherapist. And those fooled some people, right? ELIZA, you could ask it questions like, how are you feeling today? And it would turn them around. Thanks for asking. How are you feeling, right? It could play simple games and sound like a therapist. But the new models are GPT-3 is incredibly impressive. And my understanding is that the current generation of models that I've only seen sort of tweets of and secondhand accounts of are even more impressive. My understanding from talking to people is that if you fool around with them enough, you can fairly quickly figure out their weak spots. But that doesn't mean that in 2, 5, or 3, or even 1 year, there won't be a large language model that will come very close to passing just the standard Turing test. But I don't think any of them are sort of on track for the embodied Turing test. The challenge there is that if you ask me, what question would a large language, or what would a system that could pass the embodied Turing test answer that a standard one can't, that's almost by definition unanswerable. If the hypothesis is that large language models will eventually be able to answer every question that you can pose in language, then I can't pose in language an example of something that they won't pass. And yet there'll be so much that they can't do. And I keep trying to think of really compelling examples. And the problem is that it's very hard to talk about the examples, especially to talk about the ones that don't just sound like robotics. So obviously no amount of absorbing knowledge from text is going to teach you to tie your shoes, right? Or pick up a glass from the table or move a chess piece. But it also won't tell you how to farm or do many of the other things that, acquire many of the other skills that people over generations have acquired. But it's very hard to articulate what's missing, and yet I think a great deal will be missing until we include these.
Yeah. So one of your big claims is that once we figure out mice intelligence, figuring out human intelligence will be not so much more work. So could you unpack this?
Sure. So it goes back again to Moravec's paradox. Animal life, metazoans, first appeared something like 600 or 700 million years ago. And, the basic forms of life that can interact with the environment sort of appeared a couple hundred million years. Well, the basis for the modern sort of groups that can interact very effectively with the world, like vertebrates and insects, appeared a couple hundred million years after that. And so over the last, many hundreds of millions of years, all these organisms, eventually, including mammals, have evolved very sophisticated priors about what is needed to interact with the world. We have sophisticated sensory apparatus and sophisticated motor systems, and they're integrated, and we can integrate mid and higher level goals and trade them off all against one another. So the idea is that there's a great deal of evolution behind all vertebrates and even all mammals. And the basic mammalian nervous system, it was already present at the root placental mammal, which appeared, of order 100 million years ago. So on top of that, what you find is that it's actually pretty easy on an evolutionary timescale for mammals to acquire even more sophisticated skills. People might not be impressed with what a mouse can do, perhaps because they don't study mice. But I think anyone who's ever been to the park is probably pretty sophisticated, at least with the sensory motor capabilities of a squirrel, right? A squirrel can jump from tree to tree. It can run along a wire, collect nuts. If you have some time, you can look up this video by Mark Rober, who's a YouTube poster, who set up a ninja course for the squirrels that were in his backyard. So squirrels are really impressive, and there's relatively little evolutionary distance between a mouse and a squirrel. And of course, the root primates are not that far off from these kinds of capabilities. I mean, sure, there's all sorts of things that are special about us, but if you trace it back, the thing that I keep coming back to, to go back to the first question, that's most special about humans, and I think is the sort of root of most of our success, if by success you mean our ability to, expand our population on the planet to over 7 billion, right? The key there is language, and language is something that evolved probably 100,000 or at most maybe 1 million years ago, depending on whether you think that, Neanderthal and Denisovan also had language. I think the idea is that there's so much that we can't do that a squirrel can do. When I say we, that I mean artificial systems can't do that a squirrel can do. And it's a relatively small, shall I say, jump from what a squirrel can do to what a monkey can do, and from there to what a great ape can do, right? It's not like we're there when we've got a mouse, but once we have a mouse, it's really not that hard to imagine that it's a small jump to a creature that can interact with the world in ways as sophisticated as a human. I'll just sort of throw out one way of thinking about it. If you look at the kinds of structures that many mammals produce, like, a beaver makes a dam, or even, birds make these elaborate nests, they have the ability to interact with the world and in really sophisticated ways. And why can't, if you ask, why can't my dog load my dishwasher? Well, partly because he lacks opposable thumbs, but I think it wouldn't, give him opposable thumbs and a couple of 100,000 years of evolution, and we'd have dogs loading the dishwasher. So I guess that's sort of the basis of my intuition.
Yeah, I suppose if the dog's ecological niche included dishwashers a lot, they would figure it out very quickly.
Exactly. That's exactly the way I think about it. That really what limits dishwasher loading is the fact that is not their current ecological niche.
Right. So let's take sort of on interacting on the world, what can we learn from neuroscience? And in the paper, you mentioned sort of two things. So architectures of non-human animals are modular and hierarchical. And they're like two design principles that are quite interesting in terms of that, which are called partial autonomy and amortized control. Could you maybe explain those two concepts and how they could help AI?
I'm going to approach it. Those are terms more from the AI side. So I'm going to actually approach it more from the neurobiological side, because I think that's really, the basis of my intuition. And I think it's helpful to think about the solutions that biological systems have come up with. The way that the nervous system is organized is that there are layers of control over everything that we do, right? And those layers actually reflect our evolution. In the beginning, right, the challenge for the very first organisms that lived on the sea floor, perhaps, or the first animals, I should say, not the first organisms. The first organisms preceded the first animals by over 1 billion years. But the first animals that lived on the sea floor, challenge really was just locomotion. There's some thought, we don't really know this for sure, but there's at least a compelling story that the beginning was that multicellular animals would die, and then other animals would sort of wander around, and that would be, those dead animals would be a rich source of nutrients. So maybe the first animals were scavengers that just sort of crawled around. And so maybe a random walk at the beginning was just the ability to locomote might have been enough, but you could be more effective. You could outcompete other animals if you could not simply walk around as a random walk, but walk around with some goal. So you needed sensory apparatus to see if you could find the next source of food. If you imagine, like, even locomotion, by the way, is pretty challenging, right? It seems trivial, but we and many others have been playing around with training simple agents to locomote in artificial environments, and it's pretty hard to get a creature that can locomote. So, but now you have locomotion combined with sensory apparatus, and now you can imagine that occasionally when they're looking around scavenging, they come across a live creature, and why not eat that as well? So now you have predation, and now the fun begins, right? Because if you have predators, now you have prey, and the prey that can escape the predators are the ones that survive. And so we now have an arms race for ever more sophisticated sensory motor control. But, if you think about what's going on there, you need some very short time scale types of control, the type that is needed to locomote on the seafloor. And then you need somewhat longer time scale systems, which tell you where to locomote and how to balance off threats versus possible rewards. It turns out that, so if you imagine that the lowest level systems, let's say, are comparable or form the basis of something like our spinal cord, right? That's enough to get basic locomotion going. But then in vertebrates, there are a series of higher structures that don't replace the spinal cord. Of course, we still have complex control in our spinal cord, but modulate it and add to it. And so that's how you get this sort of hierarchical control over the course of evolution, where you have higher structures in our nervous system that interact with and under some circumstances control, but never completely supplant the lower structures, in many cases, evolutionarily earlier structures. And ultimately, right, in mammals, the most recent ones of those structures is the neocortex, the structure that endows us with, or that we think endows us with, high-level intelligence. But that structure is building upon all these earlier structures. So you get this modular, higher-level control. I want to add one more thing, which is the modularity is also useful because in order for evolution to proceed quickly, it has to operate the same way that a program does, right? If you write a computer program, you learn that it's much better to have little modules than to write one big program. And in the same way, having modules in the nervous system is part of what probably makes it easier to evolve rapidly along certain dimensions. So I think that's some of what we can take at least at the global scale from looking at the organization of the nervous system. Now, what I just described are sort of very high level observations, and how to take those observations and turn them into specific, realizable systems we can use to build a better AI system. That takes a lot of work and engineering and trial and error. I'm often, going back to the work of Jan LeCun. I mean, I think that's really one of the best examples where, I'm sure you know, Jan was inspired by the work of the physiologist Hubel and Wiesel, who worked on the organization of the visual system. So Jan LeCun sort of took those observations and used them to form the basis of convolutional neural nets, CNN. But it wasn't simply, he didn't simply make a model of Hubel and Wiesel. He took that as inspiration and then asked himself, well, how can I take those ideas and incorporate them into a system that will have some added capacity? And I think that's sort of how we should think about it.
Yeah, I think this bridges quite well to like a lot of the other themes in the paper, because there's a lot on NeuroAI history and also sort of on aspects about training the next generation, because people will need the engineering expertise, as you said, but also, I guess, neuroscience, but also, I guess, evolutionary expertise, which is often kind of not explored that much. And there's some other interesting things about sort of energy efficiency and spiking neural networks, neuromorphic computing, and also some issues about like grant funding and how sort of systems of financing and sort of organizing science. There's a lot about that, but I actually wanted to focus on a very specific aspect of the paper, which is sort of about this platform you mentioned. So it's a shared platform that in the future may allow the developing and testing virtual agents. What is the plan with this platform? I guess it's not very concrete yet, but sort of what are some of the ideas in that?
Yeah, as you say, it's not very concrete in the two fields. Now, in terms of the specifics of this platform, we certainly as a group haven't converged to what that would look like. And it might not look like a single platform, right? But at the same time, it is a call for at least one platform, or at least that has the scale and is sufficiently developed that we can all play on it. We can all sort of use it to drive advances. I don't really want to say too much more about what that's going to look like at this point, because, different authors have different ideas of what that will look like. I don't think that we're disagreeing, but I don't think we've fully reached a consensus on that. And it's probably not the case that all of the authors would be on a single project or anything like that. That seems highly unlikely to me.
So on this platform, it might not be one platform, might be many different platforms, and probably have loads of different tasks. And you've done some really important work on sensory decision-making rodents, where you created two alternative choice tasks. And in the past, people thought, oh, well, primates can only do this, rodents are not smart enough. And then, well, I guess you learned quite a lot from doing that. And I was wondering, what are maybe some of the takeaways in trying to like quantify behavior or progress with, well, real animals, and how that could translate to in silico agents on a potential platform or many?
Yeah, that's really interesting. I started off my time here at Cold Spring Harbour along with Zach Main in trying to figure out how to train rodents to perform complex decision-making tasks that prior to that people had only trained non-human primates on. And what's funny is that the tasks themselves at some computational level are typically actually kind of trivial. So an example of the kind of task that my lab trains rodents on is there are 3 so-called nose pokes, little holes that the animal can stick their nose into. And the animal sticks its nose into the center port that triggers a computer to deliver a stimulus. And we've trained the animal to go left for a low stimulus and right for a high stimulus. And I've just described that task in 3 sentences, and it sounds trivial. And in fact, in some sense, the decision-making portion of that task is trivial. Go left for low, right for high sound. And so the question is, why does it take a couple of weeks to train a mouse or a rat to perform that task? And why does it take 6 months to train a monkey to perform perhaps a somewhat more complex visual task. So I'll say a couple of things about that. The challenge of that task is not the part of the animal hearing, being able to distinguish a high frequency sound from a low frequency sound. It's being able to train the animal to produce a particular motor action in response to that. And he has to discover that over the course of training. I'll just say there's a funny story. It's perhaps an apocryphal story. A couple years ago, I tweeted out whether anyone could actually provide a basis for this. And I haven't found anyone who actually can support it. So I'll tell the story. Even if it isn't actually true, it could have been true. The claim is that, so as many of your listeners might know, people train monkeys to perform complex tasks that involve putting visual stimuli on the screen, on a TV screen, and then having monkey indicate its response by moving its eyeballs, by visually saccading to a different point on the screen. And it can, as I say, it can often take 6 months for an animal to learn this task. So people at some point took a bunch of supposedly Caltech undergrads and asked them to learn the task without verbal instruction. You sit in a chair and you look at a screen and you're rewarded based on what you do. But there's no prior instruction that the rewards have anything to do with, for example, where you're looking. And the claim is that Caltech undergrads were actually slower at learning the task than monkeys. So the key to rapidly being able to acquire these tasks, in part, is probably language. So that's all a way of saying that one thing that I learned over the course of this is that what's hard about interacting with the world is not the part of the task that we tend to focus on, which is, in a trained animal, how does the animal make a decision to go left or right? Now, that said, I think that that's still a very interesting question. And in this highly restricted domain, we did learn a bunch of things. We, in many other labs now that study those kinds of tasks, we learned about how animals deal with uncertainty, for example. And we learned about how different brain areas interact and communicate, how information is shared. Those remain unsolved questions, but it's key that the ability of a sensory area to share its information appropriately with other areas is central to this. And out of all the sensory stimuli that an animal is getting at a particular moment, the question is, how does it know which is relevant, which isn't? Those are the kinds of questions that when you actually deal with real animals interacting with real world problems, you realize that those become the central problems and often not the kinds of questions that we've been focusing on, which is, subtle, important, but still in some sense secondary questions about how you distinguish a cat from a dog based on a visual input.
Yeah, that's really interesting. So I was thinking with the undergraduate examples, because if you told them what the nature of the task was, they would solve it immediately, right? And I guess
they get it in 3 minutes. Yeah.
Right. And I guess you could say that with a lot of current AI work, it's similar in the way that you are like specifying the task structure very explicitly. And in a way, well, what might be important for this platform is to actually have it in a way, I guess, more open-ended. I guess part of that could be being naturalistic.
Exactly. So I think that's exactly the kinds of questions that we're going to be starting to get at. If you want autonomous AI that can interact with the world, right? For example, one of the key questions is how do you balance off the multiple goals you have, right? Not just multiple sources of sensory information, but at any given, imagine, for example, the challenge in a self-driving car, right? In a self-driving car, right, there are a bunch of goals: get the driver where he needs to go, don't crash, follow speed limits. How do you balance out those various things? I think that, so I know nothing about really the guts of how the current systems work, but I do know that contrary to predictions from a few years ago, we're really not there yet, right? And these are the kinds of challenges that biological agents solved or had to solve in order to interact with the world. And these are the ones that I think we can really begin to grapple with in sort of a richer environment where we have embodied agents.
Yeah. I'm a big fan of the work of Paul Cisek, who you also mentioned in the paper. And I guess
Absolutely.
he has the affordance competition hypothesis or something along those lines. And I guess there's a lot with animals on action selection and what actions do they choose. And I guess a lot about that might be really interesting for this sort of work.
Absolutely. Yeah, no, I think that's exactly right. Those are the kinds of issues. I mean, he's been a pioneer in this. And there are lots of other people. I should say, there's a citation limit. So, we could have cited 10 times as many groups and papers, many of whom I should really clarify. The point of this wasn't that we were the first to come up with this idea. This echoes the ideas of many, many other people from diverse fields. And we were not able to cite all those because there are citation limits, not in the bioRxiv preprint, but this is now a submitted document. So absolutely, those are the kinds of issues that many other people have been thinking of. And we just hope to be able to sort of galvanize the community to have sort of a concerted focus on these kinds of questions. So I think that's absolutely right.
Yeah, I'm not sure whether this is your area, but I find it very interesting. So I guess part of what you would want the agents to be able to do is explore on their own. And you mentioned sort of a paper which compares like a reinforcement learning agent with children. I'm not going to get kind of into details with it, but one interesting thing was that children naturally do a lot of exploration where they just look at sort of the order of events. So it was a weird task where they had to put objects on a machine. And they kind of just played around with, well, in which order do I put the object on the machine? And does that make a difference? And I thought that was really interesting because a lot of rodents research focuses on, well, can these rodents learn the order of stimuli we present to them? So if it's sounds or odors, well, I guess it makes sense in sort of this embodied sense that agents really have to make sense of, well, if I do this, what does it cause? And what is sort of the temporal order of events? And how does it make sense of causality in that way, right? Do you have any intuition on this, how this could be sort of related to exploration or could be important for the platform? And I know it's quite open-ended.
Yeah, that's a really interesting set of questions. My intuition is that I have a couple answers to that. So I guess I'm going to say something that puts me at odds with at least some people, which is that my intuition is that we should be paying attention right now to what simpler animals are doing, what non-human animals are doing, before we start thinking about the things that children do. And this goes back once again to Moravec's paradox. So I think exploration is key, and all animals do some exploration, but the kinds of exploration that, let's say, mice do is of a different, or squirrels, is of a different nature from the kinds of exploration that human children do. Although eventually, I think we would like to have a system that takes its inspiration from what children do. My idea is that I don't mean it's unique to me, but I'm saying that it might put me at odds with some people, is that we should right now derive our inspiration from the animals to lay the foundation for doing what the humans do. So exploring the sequences in which we perform actions, I think will at some point be really important and really interesting. But the first thing is to be able to perform an action, to understand how to chunk your motor commands into actions, right? We don't even know how to do that, right? The learnability of something has to do with the simplicity of the building blocks that you're putting together. Essentially, what you already have and then what you need to add to it. So you can't start thinking about the sequence of placing objects, let's say in Tower of Hanoi, until you can actually pick up the little rings that form the Tower of Hanoi. And I guess I keep coming back to that once we understand how to chunk actions at all, then we can start thinking about the flexible ordering of them.
Yeah, on that. So I was thinking we could move this to the innate learning debate. And you got a great paper called A Critique of Pure Learning and What Artificial Neural Networks Can Learn from Animal Brains. And I was just wondering whether there was a great example where you sort of made a case with cognitive maps that a lot is already innate and already only some aspects of that cognitive map are learned. And maybe you can just sort of outline that for us and maybe how it links to rodents, because I guess spatial navigation is something which is exactly what you're getting onto, is quite a lot of animals do it. Yeah.
Exactly. Yeah. So the point of that paper was that in conventional AI, at least a couple of years ago, the basic approach was to learn everything from scratch, right? So the tools, I should say the paper was written a couple of years ago, and it's actually worth thinking about how things have changed since then. But at least circa, 2017, 2018, when I wrote the paper, the standard approach, the almost universal approach, was to take a very large data set, squeeze all the structure out of it, let's say with backpropagation, and then start working with your final system, right? And then when you wanted to solve a new test, you do the same thing. And that is not how biological organisms learn, right? Biological organisms come endowed with very, very strong priors about how to interact with the world. And this is often lost on people who think about humans, because indeed, human infants come out helpless, and they do kind of seem tabula rasa, although even that's not true. But if you look at an insect, it comes out of an egg, well, at the appropriate stage of development. A spider is born and comes out ready to hunt. A fish is born able to swim. Actually, there's a somewhat gory but compelling video of a whale being born if you search it up on YouTube. And the whale is born, and the moment it comes out, it's swimming, right? And that sort of makes sense, because if it weren't the case, then it would be bad news for whales. And a colt, a baby horse, is able to stand the moment. So all of these are examples of tremendous innate ability. In particular, as you say, there of course is a role for learning in all creatures. Even insects are able to learn. And I think the idea of a cognitive map is a great example. So as you know, the hippocampus forms a map of its environment in any environment that you put an animal in. So you can record place cells in a mouse or a rat, and those place cells will differ depending on, whether you put it in one maze or another, or this cage or that cage. But the interesting point is that it is the hippocampus, this very specific structure, that always acquires that map. And the very first time that you take a rat pup and put it into an environment other than the one in which it was born, it will immediately acquire place cells. It will immediately acquire a map of that area. So the ability to acquire a map is innate. The details of the map, of course, have to be dependent on the environment, because each animal is going to be in its own set of environments. And so I think that's exactly the right way to think about it. What is innate is a set of priors for acquiring the right information and storing it, the right information from any specific environment.
Yeah, I think it's interesting. I was thinking a lot about complementary learning systems theory today. Just the idea that, well, you have the hippocampus, which is sort of learning quite quickly. Sometimes it's called like a rapid sequence generator. And then the neocortex, it kind of learns across those episodes in the hippocampus, and then does that more slowly. And I was just thinking it's interesting, sort of from this, going back to this evolution point, that, well, maybe, the hippocampus is quite conserved across species. And I guess that makes sense developmentally, because, well, there's more space to grow on the cortex across species. But I guess what's interesting is that, especially with looking at cognitive map, we can learn a lot from looking at a lot of other animals, and it'll be very similar. Whilst then, maybe if we think of the slowly learned dynamics by the neocortex, that might map better onto sort of, well, what ecological niche is this animal learning for? What is it doing with the cognitive map? What is the content that is then incorporated? And I guess there's still like a large prior, in a sense, in what is the architecture of that neocortex, because that then defines, well, how is this cognitive map used, and how you'll have different cognitive maps depending on if you're a bat or if you're a rodent, because those are completely different ways of using that cognitive map.
Yeah, it's an interesting question. I mean, on the one hand, I confess I don't really know the degree to which the hippocampus is conserved across different mammalian species. Other people know it, and I'm going to look it up after this. So it's a great question. Certainly, there are differences in the neocortex across mammals. But what's interesting also about the neocortex is that the basic structure of the neocortex is highly preserved across all mammals, right? The basic so-called six-layer structure of the neocortex is preserved. In fact, the sort of working hypothesis that So in my day job, I'm interested in cortical circuitry, and the working hypothesis in my lab and many other labs that study the cortex is that there's basically a canonical cortical circuit that is then replicated over and over again with tweaks, with modifications, but the basic circuitry is maintained. And that basic module turns out to be scalable. And that's why the neocortex can grow so dramatically across different, from mice to primates to apes and humans, right? That really the key is that the neocortex happens to be or evolved to be sufficiently modular that it makes sense that it's useful to have it grow by a factor of 100 or 1,000. So most brain structures don't have that property. Most brain structures don't necessarily scale well. And that's not typically, nuclei, which is sort of the other basic structure. They tend to be highly specialized. And it takes a lot of tweaking to modify one nucleus to adapt it to, when I say tweaking, I mean over evolutionary time, to take one nucleus and have it evolve into something else. So I think there's an interesting question about the degree to which the neocortex of different mammalian species is specialized for their niche. And to some extent, obviously it is, if you think about visual processing, et cetera, and to what extent it is sort of a generic structure. And what's special about it is that it's so easily, so modular and so easily expanded.
Yeah, I think I would, on my point, I'd say that I totally agree, like, especially on the micro and meso scale, on the neocortex, a lot is conserved. I guess, like, my follow is mainly related to the macro scale and how that organization might depend on the ecological niche. But let's move to your work on the genomic bottleneck. And this might be quite flattering for you. So I was listening to a brain-inspired podcast episode with you and Doris Tsao, and she called your genomic bottleneck Mars fourth level of analysis. So I thought that was really cool. Yeah, but I think as a starting point, we could talk about the sort of the genome and connectome, and you can give us some intuition how big it is. And I'd like it for these three species, a mouse, a human, and a marbled lungfish, because you did mention that in the 2019 paper. And I guess I'll put a Wikipedia link on what a marbled lungfish is in the description if someone wants to know the sort of details of that.
To be honest, I have no idea what a marbled lungfish is. It's a kind of fish, I guess, with marbles. So the basic idea of the genomic bottleneck is the idea that, if you take seriously the idea that we are born, that all organisms are born with innate knowledge of the world, that innate knowledge is instantiated in the connections in our brain. That's in our nervous system. That's mostly where that innate information goes, into the wiring diagram. And so where does that come from? That ultimately comes from the genome, right? And the genome is just a string for the non-biologist. It's just a string of nucleotides. There are 4 ATGC, and each cell of your body has more or less identical string of these nucleotides. And your genome is what makes you. And, the reason that when a dog is born, it looks like a dog and not a human is because there are differences in its genome from our genome. So that raises a question. How much information is there in a genome? And the nice thing is that's an easily answerable question. So we can measure the length of the genome. People do that through DNA sequencing. And the length of the human genome is about 3 billion nucleotides. And each nucleotide encodes 2 bits. So there's an upper bound to how many bits of information, literal bits of information in the Shannon sense, are encoded in our genome. It's 6 billion. It's actually a lot lower than that because there are lots of repeats and whatnot in our genome, but it for the purposes of this, it doesn't matter. And mammalian genomes are all roughly the same size. I don't know off the top of my head exactly how big a mouse versus a human is, but it's actually it's not at all intuitive. So you might think, as you get to a more complex organism, you get a longer genome, but there's no intuitive notion of complexity that for the most part, by the time you get to multi-celled animals, that really works out for. So, mice and humans have, and most mammals have genomes, I think, that are in the same range as the human. Humans are typical for mammals. As I mentioned, I looked it up, and at least at the time I wrote that the record holder among animals was the marbled lungfish, which has a genome that is more than an order of magnitude larger. And the point is that there's really nothing obviously special about a marbled lungfish that would explain that. In fact, my suspicion, I don't quite remember if this is true, is that it might have to do with long repeated sequences due to transposons, which is another subject. So there's nothing special about having a huge genome. The only reason I mentioned that was that you might think that the reason we don't have a bigger genome, and therefore the reason that we don't encode more about our nervous system, is that we can't fit a larger genome into our cell for some reason. But that's clearly not the case. There are animals, like I say, that have genomes that are 1 or even close to 2 orders of magnitude bigger. So we could, if we needed to, in the evolutionary sense, if we needed to, have longer genomes. And so the hypothesis in that paper is that the reason we don't have a longer genome is that it's actually a feature, not a bug, that the rules for wiring up our nervous system, that the innate knowledge that we carry from generation to generation is actually squeezed every generation through this genomic bottleneck, because that squeezing process is can be thought of as a constraint, and constraints are actually what will encourage generalization. So by forcing the nervous system to by forcing the rules for wiring up the nervous system to be very simple, as they must be if they're going to fit in our genome, what evolution has really done is it has put strong pressure to encode only those things that are useful and not those things that are not. And that is how you can encourage generalization. So that's sort of the core idea, that the genome acts as a regularizer.
Right. So with Alexei Kulakov and Sergei Shurave, I hopefully pronounced that name not too badly, you sort of have concretized this in a paper last year called Encoding Innate Ability Through a Genomic Bottleneck. And could you sort of just describe the genomic bottleneck algorithm for us and how it compares to transfer learning, which is an active area in AI?
Yeah. So the idea of the genomic bottleneck is actually very simple. It's inspired by what I just said, that really what you want to do is encode the rules for wiring up a network in some simpler way. And so what we do specifically is we train a network on actually several tasks, including standard MNIST character recognition and CFAR image recognition, and then more recently on some basic reinforcement learning tasks. Then we take that trained network and we take a second network that learns the structure of the first network. And the particular way we do it, the details of it aren't that interesting for the purposes of this podcast, but we basically have a smaller network that spits out an approximation of the original network of the trained network. And of course, so basically that can be thought of as a form of compression, of lossy compression. And we iterate this process until we discover those, we call them phenotypic networks, P networks. Those are the ones that perform the task. We discover P networks that are both compressible and solve the task pretty well. And so eventually what we get is an initial performance that is encoded in the genomic network. So we end up with a genomic network that is much smaller than the phenotypic network, and the initial performance of the phenotypic network produced by the genotypic network can be really quite close to the final performance. So with basically a network that's 1 or 2 orders of magnitude smaller than the final network, we get performance that's quite close to the final performance. And that's sort of conceptually like what we have in animals, where an animal is born almost ready to interact with the world. It still needs to learn more about the world, but it sort of comes close. So that's the inspiration, and this is just the first stab at it. There are many other ways one could think about compressing the world. In fact, around the same time that we put this out, Wolfgang Maas sort of took the same idea from the critique of pure learning paper, came up with a somewhat different way of compressing networks that I found really interesting. And there are, I think, several other approaches. But how does it relate to transfer learning? I mean, in some sense, you could think of it as solving, there's transfer learning, there's meta learning, there's continual learning. All of these are sort of in the same basket of ideas. They're different problem formulations. I think what we do is different is we're trying to spit out the entire network overall, whereas many of the transfer learning approaches often sort of have a different approach to how the final network relates to the initial network. And I should just circle back and say that I think in a lot of ways, in the same basket of ideas that these so-called foundational models for, let's say, foundational language models are found. And it's sort of addressing the same set of problems, which is that you don't want to keep relearning on an entire, the cost of training a large language model these days is in the millions or tens of millions of dollars. And so even Google can't afford to keep spending $10 million each time they want to try something new. And so instead, what has happened in the last couple of years is that you train one of these models once, and that becomes the foundation for a whole host of related models.
So one interesting thing in the paper was that, and this sort of relates to the visual system in animals, is that when you took the sort of the first lower levels of this genome network and you used it to then try to learn a new data set, that did really well in terms of, I guess, the transfer learning air quotes. But if you did it for the last layers, that wasn't the case. And could you sort of explain the intuition behind this?
Yeah, so I mean, these are just observations, but our intuition about what's going on. So one interesting thing we found, I'll just step back, is that when we performed this on the simplest system, MNIST, character recognition, it really didn't work very well at all. And I think that's because the structure of MNIST is so simple that it's still easier for the genomic network to just essentially find a way of memorizing that whole data set in some way. When we trained it up for images, indeed, what we found was that the earlier layers of the network seemed to be doing some kind of early visual processing, which is general, and the later layers of the network were specialized for the specific task that we trained it on. And I think that it sort of has the same flavor that we find in the visual systems of different animals, where if you look in the early visual system, there's a lot of shared structure in mammals, but we don't find face cells, which are later in the visual system. We don't find face cells as far as we know, in rodents. Actually, may turn out that they do learn a little bit about faces from each, but we don't know that yet.
Right. So what you sort of say in the conclusion that it's a bit similar to what you're doing with the algorithm to Lamarckian evolution, and Lamarck is obviously laughed upon because this sort of idea of, well, if a giraffe reaches the fruits on a very high tree, they'll have a longer neck and it passes that experience through to the next generation. But in a way, that's sort of what you're doing because you're compressing the learned network then onto the G network. And I just thought it was interesting sort of returning now to the platform and thinking how evolutionary things can be implemented into it, because I guess there would be reasons for doing that, because then I guess this also slightly relates to what you call supervised evolution in the 2019 paper, and this idea that, well, you are by artificially compressing the learned information, that's more efficient than just trying to compress in a way the wiring rules, which are the ones that are specified in the genome, and that then there's selection. But so what are maybe some of the reasons why actually not doing that and sort of doing maybe Darwinian evolution-like implementation in the platform would make sense? Or do you have sort of intuition on like weighing these up?
Yeah, I have strong opinions about that. So obviously in biology, what we have is Darwinian, not Lamarckian evolution. Lamarckian evolution, as you say, is the idea that traits that you've acquired in the course of your lifetime can be passed on to the next generation. That seems very intuitive, but it's just not how things work. And one can ask, why isn't it why things work? So my answer is that Lamarckian evolution is a much better algorithm, and that's how things should work. But that isn't how things work. In fact, I think Darwinian evolution corresponds essentially to a random walk in parameter space, in the parameter space of your genome. And that's a terrible algorithm. Pretty much all significant advances in modern AI and machine learning come from exploiting the gradient, come from basically doing gradient descent. I mean, to my knowledge, there's almost no successful algorithm that involves lots of parameters that doesn't involve one way or another exploiting a gradient. And evolution is able to get away, Darwinian evolution is able to get away with not using this gradient through brute force, right? So I did a back of the envelope calculation about the number of animals that has ever existed. And it's hard to get the numbers right, but the ballpark is at least well over 10 to the 30 lifetimes of individual animals since, 500 million years ago when the first animals evolved, right? That's a huge number. That's so many more. That's 10 to the 30 lifetimes, maybe even 10 to the 35, maybe 10 to the 40, who knows? And so even if you have just an awful algorithm, like wandering through high dimensional space, with the brute force of evolution, you can still come up with real complexity. But even though computers are very fast, they're nowhere near fast enough to be doing that, to be working on that scale. And so what we're calling Lamarckian evolution is basically a way of exploiting the gradient. We learn from what we got in a previous generation, and that helps us figure out where to go for the next generation. And so that's, I think, the key intuition for why I think anything that we do in this sort of engineering setting is going to have to, if it involves, a large number of parameters, as it likely will need to exploit a gradient and therefore use something like what we're calling Lamarckian evolution.
Okay, so my next heading is Connectomics. And I know that maybe I don't know, but the intuition is that a lot of people, neuroscientists, don't care about neuroscience too much, but they especially don't care about architecture. And why should people in AI care about wiring rules, developmental processes, and connectomics specifically?
So we know that the structure of a network, an artificial neural network, essentially endows that network with a bunch of priors, right? And basically the solutions that you find are a combination of the training set, the data that you get in, and the architecture / learning algorithm that you use to fit the data in a supervised context, right? And so the structure of the network basically instantiates some prior about what you're going to end up with. I go back to the great example of a convolutional neural network, which is a network with a very specific structure, and that instantiates a very specific prior about translation invariance, right? That was the insight that structure captures a prior about translation invariance, which is a property of images. That's why they work. So it raises the question, are there other interesting priors that one might want to endow a network with? And one approach to that might be to search through the space of all possible networks. And I don't think that's an unreasonable, come up with the clever ways of searching through the space of all possible networks. I think that's a reasonable approach. But another approach is to actually double down on the insight that we got from Cubel and Weasel and the structure of the visual system and ask, are there other rules, other sort of priors that we can get from looking at the actual structure of actual nervous systems? That then raises this issue, which is that we don't actually know the detailed wiring of many nervous systems. We understand the detailed wiring of the worm C. elegans, but that's a worm that has 302 neurons, 7,000 synapses, and is highly specialized for a specific set of tasks. We don't really know that much about the wiring of the mammalian nervous system. This is sort of my reformulation of the motivation. My real motivation for starting to get interested in the wiring of the mammalian nervous system had to do with my day job, which was to relate neural activity to behavior in awake behaving animals, which is some of the work I talked about earlier. And about 2009, 2008, 2009, when I was sort of working, trying to tease apart the circuits underlying auditory decision making, I realized that it was very difficult to figure out those circuits without having sort of a good guess as to the overall structure of those circuits. And at the time, there was growing interest in using electron microscopy to figure out the wiring diagram of circuits. But the challenge there was and remains that EM, electron microscopy, is a very, very, very slow technique. And it has now gotten faster by a couple orders of magnitude, but it remains a very slow and expensive technique. And it wasn't obvious to me that it would be easily scaled up or even eventually ever scaled up to figuring out the detailed wiring diagram of a mammalian system. And so I had the idea of converting the problem of the wiring diagram in a mouse or a rat into a problem of DNA sequencing, just because DNA sequencing, for those of your listeners who are not biologists, DNA sequencing has just exploded at this incredible rate. Like the rate at which the cost of sequencing has been plummeting at a rate that, at least for a while, was faster than Moore's law. And so sequencing millions and billions of nucleotides can be done for, tens or hundreds of dollars at this point. And so if we could convert the problem of circuitry into a problem of DNA sequencing, we would be able to potentially have the scale and the throughput to actually figure out the wiring diagram. And so that has really been sort of one of the primary focuses of my lab for the last 10 plus years.
Yeah. So you, just as Capsule and a few other people, well, you're revolutionizing connectomics. And one of the important papers was the 2016 paper on MAPseq. And could you sort of just explain what that is and how it works?
Sure. So the core idea is that what makes figuring out connectivity in the brain complicated is that neurons have these very long axons. So a neuron is a little structure. Its cell body is maybe 20 microns across. It can have an axon, which is how neurons talk to each other, that can be that can be thousands of microns, millimeters, or in the case of a giraffe, meters long. And each neuron can produce, send out thousands of these processes, and is connected to thousands of other neurons. And it just looks like a mess. It's just a bunch of wires. It's just a bunch of spaghetti when you look at it under the microscope. And trying to trace the detailed connection from one neuron to another is incredibly challenging. The traditional solution, which has been going on for, decades and at this point even centuries, is to fill one neuron at a time. But, a mouse brain has 100 million neurons. And so that's a lot of mice if you want to do it one neuron at a time. Plus, each mouse brain is different. So it doesn't really tell you much about connectivity. So there was really the inspiration for my approach came from brilliant paper by Jeff Lichtman and Josh Bain, first author Jean Livet, which was that it's much easier to follow the trajectories of individual neurons if you give them different colors. If you imagine, when you open up, a device and see the red wire or the green wire, you have to know which one to cut. It's really convenient if the wires have different colors. And so they figured out this very clever genetic trick for giving different neurons different colors. It involves sort of genetic recombination. The problem was, even though the genetics of it worked out brilliantly and they could come up with a palette of over 200 colors, unfortunately, because of the nature of how these colors are generated, they can't really be practically distinguished. So the colors are generated by combining fluorophores of red, blue, and green in different levels. But even though they could potentially have 200 different combinations, you can only resolve maybe 3 or 4 or 5 on a good day. It makes for beautiful pictures and there's some niche applications, but it just hasn't been that practical. So the big idea that I had was if we could replace colors with sequences, we now have an infinite number of sequences of DNA. And so the first graduate student I managed to convince to work on this was Hassana Oyibo, who pioneered using, developing these DNA barcodes in neurons. There was another grad student, Ian Pycon, who figured out how to take those barcodes and drag them to synapses, fill axons with them. That in itself was a challenge. And it was Justus Kebschel, who was the author of that 2016 paper, who first was able to apply it in a way that was useful for teasing apart the connectivity of a circuit, or I should say the projection patterns of neurons in a circuit. He figured out how to take the system that Ian had engineered and actually use it to look at the projections of the locus coeruleus, and then later in collaboration with Tom Mersick-Vogel and others, to look at the wiring diagram in the visual cortex of the mouse. And over the last few years, it's been applied both in my lab and several other labs by now. I would say probably, I should do a count, but it's probably at least a dozen other labs at this point, to a lot of other areas of the nervous system. And I think it's really beginning to catch on as a useful tool for large-scale interrogation of the structure of the nervous system, both in mammals and even in other model organisms.
Yeah. So you mentioned earlier that one of the problems was also that if you, with the classical method, that you have to use different mice and they'll have different brains, and then just the co-registration process of that is really difficult. And you came up with something new called BRICseq. And I just want to sort of quote it because I find comparative connectomics very interesting. So the last words of the paper are opening up the possibility of a new era of quantitative comparative connectomics. So that's really exciting. And a lot of that connectomics work is done with diffusion MRI right now. And sort of what are some of the advantages and disadvantages if you compare those two methods that are especially important for comparative connectomics?
Yeah, absolutely. So I'm really interested in this comparative connectomics. In fact, so Justus Kebschel was a grad student in my lab and now has an independent lab at Johns Hopkins. And this comparative connectomics is really the focus of his lab. I think that diffusion MRI and our technique are pretty complementary. Diffusion MRI gives you, and I should say I'm not an expert in it. I've read a handful of papers, but it's really not my area of expertise. But I think it gives you fiber bundles, and that's useful. But what we get is detailed single cell connectivity. So we get to look at the connectivity of thousands or even hundreds of thousands of neurons in a single brain. And we can say with increasing spatial resolution where each of those 100,000 or 200,000 neurons project throughout the brain. So we get a much richer picture of the connectivity of the brain. The other thing that we're doing more recently is we're combining it with information about the genetic identity of a given neuron. And it turns out that, looking at the transcriptome of a neuron, the genes that it expresses, tells you a lot about its developmental origin, its function, and a lot of other properties. So by combining the genetic identity and connectivity of individual neurons, we're learning a lot about the structure of the circuit. And the thing that I'm really excited about in the lab is most recently, we've developed a pipeline for combining those two sources of information, the connectivity and the genetic identity, with functional information obtained using two-photon microscopy in animals that are performing various tasks. So we get to say that, this, neuron 27 would fire when the animal heard a high frequency sound and as it was scratching its nose. And then we get that kind of information for thousands of neurons. And then we can combine that information with the information that, neuron 27 also expresses this complement of genes, indicating that it's of a particular cell type, and it sends its connections to brain areas X, Y, Z, A, B, C. And so we're getting like these just massive data sets that are just a joy to sort of work through and see what kind of structure we can extract from them.
Yeah. So this is one sort of a personal question. So I've got a research interest in comparative connectomics, maybe like for my master's or PhD, particularly in corvids, because I think they're super cool. I might put some like references in the description. But so I like the idea of looking at a lot of species in a taxa that are quite close and then sort of comparing behavioral markers in phylogenetic distance and like how that compares to maybe anesthetized, not in a task, but just like resting state. And I was thinking the BRICseq work is really cool because, well, you don't have the problem of needing to do loads of having a lot of animals per species. But then also I was thinking, well, you have these injection sites and you sort of, do you need a lot of anatomical knowledge? Because with birds, there's very little work done compared to, let's say, rodents or primates. And how easy do you think, sort of with the quote of a new era of comparative connectomics, like, how much work do we need before we can effectively use BRICseq, or what's sort of your intuition about this?
Yeah, those are great questions. And I should say, if you're interested in this, you should definitely reach out to Eustace, because this is exactly the kinds of stuff he's doing, as well as some stuff in my lab. So one nice thing about these approaches is that you don't need a huge amount of prior knowledge about where to do injections. The reason is that the spatial selectivity in these approaches derives from the later dissection, and also because we're now scaling them up into a purely microscopy friendly method. And so depending on exactly what one wants to do, one can actually be agnostic as to the brain structures involved. Still early days, and the experimental design, of course, is a complex question that requires sort of looking specifically at a particular model system. But those kinds of, that's exactly what these kinds of techniques are useful for. I should add that, or should mention that one thing we're doing in my lab in collaboration with Arka Banerjee, whom I mentioned earlier, is, so I mentioned that he works on the Costa Rican singing mouse, and its brain is very similar in size and overall structure to the lab mouse, even though evolutionarily they're fairly distant, but they can't interbreed or anything like that. But the brains are they look very similar. And the lab mouse doesn't sort of engage in these complex vocalizations. And Arka has, in his postdoc work, worked out some of the circuitry that the Costa Rican singing mouse uses to vocalize. And so right now, what we're doing in collaboration is we're comparing the two species to see sort of what we can learn about the connectivity in the Costa Rican singing mouse that sort of is responsible for this gain of function for vocalization not present in the mouse. And it's still early days, but it's looking pretty interesting.
Yeah, I'm looking forward to when that is out to read that. So I have two areas of questions left. So the first is sort of, I guess, a bit sociological, you could say. So you were on an online forum discussion called The Learning Saloon a couple of years ago, and you presented some of your work, and there was an interesting sort of discussion across the panelists about your claim, how much can figuring out mouse intelligence tell us about human intelligence? So there was this, I guess, dynamic of how much do we believe in evolutionary continuity? And then also back to the innate versus learning debate. Do we need a connectome? Is it just about the magic source learning rule? And I thought maybe not necessarily what is the right answer, because I think we know your views on it, but what do you think motivates maybe these differences in beliefs, maybe also in sort of a sector sense of neuroscience of AI, or is it sort of, do you think it's other aspects? What could maybe cause it?
So that's a really interesting question. If you'd asked me when I started college what I thought, as I said, I ended up in linguistics because I thought that we're special, and that I didn't really see much continuity between what we can do and what animals can do. I wasn't very interested in, I had a dog. I liked playing with my dog, but I wasn't really motivated by studying animals. I think that my intuitions come from following a biologist and neuroscientist path. I think most biologists, and I think many neuroscientists, with the exception of those neuroscientists who spent their career studying humans, I think most biologists and non-human neuroscientists share my intuition that there's continuity. That's sort of one of the almost central tenets of biology is that unless proven otherwise, things are conserved over evolution. And even when they're not, right, you can see how this new ability or structure evolved out of the older one. So I think that's where my intuition comes from. And I think that people who don't share my intuition doesn't mean they're wrong, although of course I think they are. But I think it comes from different experiences. I think if you just look around at your daily experience, you're surrounded by other humans who can do many more interesting things than animals. And then you look at children and, the helplessness of infants, and you have this intuition that this infant is a blank slate. So I think it's a really interesting question, how people derive their intuitions, which often sort of follow them through their whole career. And I think that is my personal path to developing those intuitions.
Yeah, I totally agree with from the sort of the biology side of things. That's probably you're working with animals, you're reading about animal models, and it's the career path you're taking, and it's sort of the existential framework of, well, this is why I'm taking this career. And I think equally one might say that a lot of sort of AI culture surrounded, I guess, sort of the breakthroughs with like Deep Blue and AlphaGo and these sort of miracles of, well, chess is like a game that is learned, Go is a game that is learned. And I guess maybe there's the cultural equivalent of, well, it must be about learning because this is where these are the examples that are talked about, right? And I guess if the history of AI was different, maybe it would be less on learning and more on innateness.
I think that's exactly right. So one of the points I made in my critique of pure learning is that learning is a misnomer, that optimization that we're doing somehow took the name learning. But a lot of that optimization is actually equivalent to what we quote unquote learn over the course of evolution. So yeah, I think that's exactly right. In fact, I think there's a lot of confusion because it turns out different communities use the term learning differently. To me, as a biologist, learning is the changes in your nervous system that you acquire in your lifetime that enables you to adapt to whatever you're born with. And there's other stuff that occurs, outside of learning. But for people in different fields, there are some who are tempted to talk about learning over the course of evolution. And I think although biologists occasionally might use that phrase, it would be purely metaphorical. None of them would be serious about calling that quote unquote learning, but that's just words. Nonetheless, the different usages of different words can sometimes impair communication across communities. So actually, it's funny that you should mention Twitter. One of the reasons I really enjoy talking to people over Twitter is that I get to talk to really bright people who are pretty far outside my own field. And one of the main things I learn is how they think about, how they conceptualize the same space of problems, and also how they use words and how it differs from how I use words. Ultimately, I don't really care what words people use as long as we can agree on their meaning. And so I often spend a lot of time sort of trying to clarify what people mean by that instead of getting hung up on, is this learning or is this not learning? To me, the words that you apply are not so interesting. It's the concepts that underlie those.
Yeah. So you are one of the co-founders of the conferences Nisus and Cosine, which are really popular now. And I was just wondering, do you think one could try to build bridges on this in terms of having an event about this with a discussion or something? And would it be then about semantics of which words are we using, or what else do you think one could cover to implement this?
So I think that, as you say, I'm one of the founders of Cosine, and Cosine is increasingly moving into the sort of, as it's everything, into the NeuroAI, into AI, and in particular, the niche that Cosine could take is NeuroAI and Nisus as well. So my idea is that the key is to bring people together so that they can start learning to talk to each other again, as they did back in the early days of NeuroAI. And I personally don't think having a panel discussion about how to use a word is very interesting. I think I think it's useful to mix people up, to bring them together, and they'll have conversations. And with time, people will figure out that, we're speaking different dialects. And, when you say there are all these amusing examples of how British English and American English differ, and some of which can lead to pretty embarrassing confusions. But ultimately, we speak the same language, and Brit spends a little time in the US or vice versa. We can figure out how to talk to each other, right? It's not an enormous gulf. It's not like if I had to learn Finnish or something. That would take a lot more work.
Yeah, that would be a whole nother career. Yeah.
Well, I do speak some Hungarian, so of course that wouldn't really give me much bootstrap for Finnish.
True. So talking about learning stuff, let's get to the career questions. And as I said, a big aspect of the Embodied Turing Test paper was about creating this new generation of NeuroAI researchers. And obviously they'll need technical expertise in the engineering aspects of AI and the neuroscience aspects of neuroscience. But what skills besides those technical aspects would be really important for this new generation?
Yeah, so there's no one right answer to that. Neuroscience is so incredibly broad that no neuroscientist knows how to do everything or even most things. But what I would say a well-trained neuroscientist, even today, even though it's an incredibly broad field, what a well-trained neuroscientist has is some familiarity with most branches of neuroscience, right? So a molecular neuroscientist these days, even a molecular neuroscientist should know the basics of theoretical neuroscience, and that most modern programs, they get some training in that. And, a systems neuroscientist these days must know a fair amount of molecular and cellular neuroscience. So neuroscience itself is broad. And what I'm trying to do is get people who have expertise in AI to get that same foundation. The way we've been doing it is we actually have a program here at Cold Spring Harbour funded by the Schmidt Foundation, the former CEO of Google, Eric Schmidt, which takes people trained in AI and embeds them in a neuroscience lab for a couple of years so that they get real familiarity with neuroscience. And typically what they'll try to do is they come here and maybe do a project in a neuroscience lab, not necessarily because they're going to transform themselves into neuroscience, but because that's really, getting your feet wet like that. That's how you can really make the transition. I don't think there's a single set of skills or knowledge in neuroscience that are required. What I envision is that people with AI backgrounds could come and learn, sort of get a general overview of neuroscience, dive into one area in detail, and maybe leverage what they learned there and bring it back to AI. I think that's my vision, at least.
Yeah, I agree sort of on the idea of levels, that you of course have like David Marr's levels of analysis, and that it's really important for neuroscience to have people to bridge those levels, because otherwise it's just, we get so far in each of them, but we don't know how to connect them. And then we're in a way much farther than we think we are, because we have no idea how to connect those different ideas. So my last question is, when do you think we will have a virtual mouse that can pass the Embodied Turing Test? So this is one of those you have to give a number questions.
Yeah, so the best answer I should give is one that makes it far enough away that I'm not held accountable for my response, and close enough that it's worth pursuing it now. So I believe that, in the same way that self-driving cars have been about 5 years away for the last 10 years, I would say a virtual mouse that can pass the Embodied Turing Test is currently 5 years away and will remain so for the next 10 years.
I like that. That's a good response. Well, this has been a great conversation. Thank you very much, Tony. It was awesome talking to you.
It's been a pleasure. Thanks so much.
I hope you enjoyed this episode and you're warmed up for Connectomics, because there will be plenty of that in the next episode. To find out when episodes are coming out, follow my Twitter account linked in the description. Until the next time.